I'm Robin May. I'm Professor of Infectious Diseases at the University of Birmingham and currently serving as Chief Scientific Advisor to the government's Food Standards Agency. We're interested in infectious diseases, in particular ones which involve an interaction between the host and the pathogen, so those that tend to strike people whose immune systems are suffering in some way, so AIDS patients, people who've received a transplant, or people who've been in a major accident and have traumatic injuries. I won the prize in 2010 and it was absolutely fundamental to the kind of trajectory of my career and my lab ever after. The biggest difference was that this prize, unlike many other research awards, is incredibly flexible, and it allowed us to move with the changing pace of science and changing discoveries over the next 5 or 6 years. So in our case, we set out to investigate an outbreak of a lethal fungal infection that was at that time very active in the Pacific Northwest in Canada and America, and could really follow our noses about what was causing this outbreak, what led some people to be more severely affected than others, and ultimately to understand the evolution of this particular pathogen in that particular setting. The really big difference with the Lister is the flexibility of the funding and the fact you can use it in lots of different ways. So in my case, I guess I kind of theoretically overspent that money 5 times because I would use it a lot to, for instance, underwrite a fellowship for other people in my lab. So they would come and say, I would like to bid for my own funding. And I was in a position to say, if you don't get it, we can still employ you. But if you do, that's great. It also allowed us to invest in new methods that in a traditional research grant would not have been written into the proposal, and therefore you could not have spent the money on. But the Lister doesn't mind about that, and so it allows you to capitalise on the latest discoveries as your research goes on. And in our case, that involved a lot of, at that time, pioneering DNA sequencing, which was something that was just becoming affordable for labs like ours. At the moment, I have a slightly dual lifestyle, so I still have a very active academic research group, and we're really interested in that continuing question of how pathogens evolve. These days, we still work in one major pathogen that we've worked on for many years, a fungus called Cryptococcus, but we've also started working on a very unusual pathogen called Prototheca, which has evolved from a green algae, so from a photosynthetic plant, to now be a parasite of humans and other animals. And then with my other hat on, I'm currently serving as a government advisor. I spend part of my week using my science in a really very different way to advise on government policy and our response to things like food poisoning incidents or international trade.